We've been through basic chemistry and we moved on into organic and inorganic compounds, and we know a little something about pH. In order to understand the living system, now we have to see how that comes together, and that comes together in cells. Cells are the basic unit of living things. They're the smallest independently functioning unit of an organism. They consist of a nuclei, organelles, and they're surrounded by cytoplasm and enclosed by a membrane. There are two types of cells that we're dealing with here, animal cells and plant. We're going to start with the animal cell. The membrane is a protective envelope that surrounds cells. It controls what moves in and out of the cell. It varies in permeability. And permeability means what is able to enter or leave the cell. There are three types of permeability: selective, permeable, and impermeable. Selective permeability is when the membrane will only allow certain things to pass. Whatever it wants in can go in or out. Whatever it doesn't want to leave or enter will not be allowed to leave or enter. Permeable, the membrane allows any substance that fits through to pass freely. So as long as it fits, it's going to pass through. It's like a channel, or if you open a door. If you open a door, anything that can fit through the door is going to come through, in or out, either way. Impermeable, the membrane won't allow anything to pass through. So if it's impermeable, it's more like a wall. You just can't get through it. So there are three types of permeability: selective, permeable, and impermeable. When we look at this picture, we see a beautiful depiction of the cell membrane. You can see that there are things in certain areas that can't get through, and in some areas they're passing freely, like these right here that I have the pointer on. They're not going to be able to get through. That's impermeable. These are passageways that go all the way through the cell, so that would be very permeable. And here you can see where the cell membrane forms a capsule, I guess you'd say, around different molecules that it wants to bring into the cell. It will do it the other way around to take things out. Here's one forming over here. So it'll move things in and out of the cell selectively. It's going to decide, okay, these can come in, these maybe can't, so it won't allow those in. A lot of the work that we did looking at lipids, talking about whether they're hydrophobic or hydrophilic, looking at the polar and nonpolar aspects, become really, really important because of this reason right here when we're talking about biology, the cell membrane. We have to take a close look at that membrane. It is made up mostly of lipids, and it has some cholesterol and some proteins embedded. But for the most part, when we look at this membrane, when we look back here, this whole area, this whole membrane is lipids. And now we'll have proteins in here and some cholesterol to kind of hold its shape. But for the most part, it's made out of lipids. And if we look at this structure right here, this is our lipid structure. When we talked about lipids, we talked about the glycerol, and attached to the glycerol, we had the fatty acid, which has a carboxyl and a hydrocarbon chain. Then we looked more closely at it, and we saw where there was oxygen, this was going to be polar, which means it's hydrophilic and it likes water. And we looked at the hydrocarbon chains, and we saw that they are non-polar. They don't have oxygen, and they're not going to be attracted to water. This becomes a really, really, really important functionality. The membrane is composed of a lipid bound to a phosphorus group. So when we talked about lipids, we talked about having the glycerol and the glycerol. I'll draw it here. Remember, and it was hooked up to 3 hydroxyls. And these were all hydrogens. I still haven't gotten the hang of the pencil, but at least we have some idea of what I'm trying to talk about here. So we'll put all of our hydrogens on here. And on this side was our hydroxyls. Remember that? Okay. So when we look at this molecule, which is the phospholipid, which makes up the cell membrane, the first thing I want you to notice is right here is where our glycerol is. Okay? Here's the glycerol. Here's this molecule that we looked at last week, right here. And we saw that when we bound this to a fatty acid, the fatty acid had a carboxyl that looked like this. And it was hooked up to a hydrocarbon chain that could be 4 to 24 carbons long. So we'll just go ahead and make a drawing of that. And it's all these valence electrons are hooked up to hydrogen. I think you get what I mean, even though my drawing's pretty bad. Okay, so there is here is the hydrocarbon chain, here is the carboxyl, and here is the glycerol. And we saw that we had to take this hydroxyl and this oxygen and remove it as water, right? That's our water. We had to pull that out to make a dehydration bond, and that linked these fatty acids to our glycerol to form a lipid. Now, in the case of the cell membrane, it is a diglyceride. It has 2 fatty acids attached to the glycerol. Now, when we look up here, do you see where the glycerol is? Let me remove this drawing. Now, when you look up here, this is where the glycerol is. It's right here. This is our carboxyl. Right here is the carboxyl group from our fatty acids. So that's the carboxyl right here. Here's the glycerol up here. Here's our 3. There's our glycerol. And here are the hydrocarbon chains. Now we know that these hydrocarbon chains are non-polar. I'm not good at this pencil thing, but yeah, they're non-polar. And we also know that if it's non-polar, what's the relationship with water? The relationship is hydrophobic. This is a real important concept to understand. Here are the hydrocarbon chains. They're non-polar, they're hydrophobic, which means they are not going to interact with water. Then we see the carboxyl group of the fatty acid right here, and its attachment to the glycerol. We're attached to 2 carbons of the glycerol, which means that we have a diglyceride. Okay? Now, if we look up here at the carboxyl, we have oxygen, so this is polar. And if we look at the glycerol, it also has oxygen. It's polar. And if something is polar, we know that it is hydrophilic. I know this is crazy, it's funny, but I'm just trying to do this real fast so it doesn't take forever. But hydrophilic, this is polar, it's hydrophilic. This is polar, so it's also hydrophilic. So that is the lipid portion. The lipid is the glycerol, the carboxyl, and the 2 hydrocarbon chains. So the glycerol is attached to two fatty acids, which makes it a diglyceride. So when we talk about our membrane is composed of a diglyceride, okay? So the diglyceride has two tails. I can't write either. Okay. Maybe by the end of the semester, I'll finally be good at this. We'll see. Okay, so now here is our lipid. The lipid that makes up the cell membrane is called a phospholipid, because the lipid is attached to a phosphorus group. And again, when you look at the phosphorus group, you see there's oxygen, so it is polar, okay? And if it's polar, it's hydrophilic. It's going to be very attractive to water. So now if we look at this, we know that this part is polar, and it's going to be hydrophilic because it's attracted to water. And we know that the tail, or this part down here, the hydrocarbon chains, are non-polar. So these are not going to be attracted to water. They're going to shy away from water. I go on and on about it because the concept is so important. It's what makes up the cell membrane, and it's what keeps cells intact. If we don't have cells, we don't have life. So it becomes very crucial to our understanding. Now, when we look at the actual chemical breakdown, we see the hydrocarbon chains, the carboxyls, we see the glycerol, and up here we see our phosphate group. So now if we look over here, here's our fatty acids, here's our carboxyl, our glycerol, and our phosphate group. Is it okay if I take and show this polar region, the carboxyl, the glycerol, and the phosphate, as just a circle? If I have just that circle, here's what I want you to know. I want you to know that it is made up of the carboxyl, the glycerol, and the phosphate group. It is polar, and it is hydrophilic. It's very attracted to water. And we will call this the head. And then we have two non-polar hydrocarbon chains, and we will call those the tail. So after this explanation, we'll mostly be seeing the phospholipid look like that. And we'll know that the head is made up of these three molecules, the carboxyl, the glycerol, the phosphate. It is polar, and it is hydrophilic, okay? And these are the hydrocarbon chains. They are non-polar, and they're hydrophobic. Very, very important. If you have any questions about that, review your lecture or get a hold of me somehow and let me know, and we'll try to work it out. All right, so the membrane is composed of a lipid bound to a phosphorus group. So it is called a phospholipid. It is called a phospholipid. Phosphate group plus a lipid is the phospholipid. That is what makes up the cell membrane, phospholipids. They have water-soluble heads that form the surfaces. They're hydrophilic. They like water. They have a polar charge, so they're attracted to water. They have a water-insoluble tail that forms the interior. They're hydrophobic. They don't like water. All they're made up of is carbon and hydrogen, so they don't have a charge. Water tends to push them out of the way. They don't want to be anywhere near water. Got it? All right, so as long as we know this head, phosphorus, glycerol, carboxyl, polar, hydrophilic, the tail, hydrocarbon chains, non-polar, hydrophobic, we're doing really, really well. Also, that this molecule is called a phospholipid. All right. Phospholipids form a double layer. So the story doesn't end there. Here's a phospholipid, and here's a phospholipid right here. But if you look, there's another one. There's a phospholipid right here. Do you see that? So when the cell membrane is formed, there's two layers of the phospholipid. It's called a bilayer. A bilayer. It is the phospholipid bilayer. Now, when you think of a cell, whether it's a plant cell or an animal cell, it doesn't really matter. It's filled with water, and on the outside is water. That's why we have to be hydrated, right? The outside is also filled with water. So here's the depiction of a cell membrane, be it plant or animal, actually. There's water on the inside of the cell. There's water on the outside of the cell. And if you look, there's a bilayer of phospholipids, because here is the head and the two tails, and here is a head and two tails. Now, remember we talked about the head is polar and it's hydrophilic. It likes water. So notice that the head is always pointing at the water, whether it's pointing to the inside or the outside. And that's how it forms this bilayer. If the tails were here, it would spin, so the tails could go to the inside. See how they're tucked in the inside? They're not anywhere near water. This is why the structure and understanding the functionality of the phospholipid becomes so important. It holds the membrane together. It holds it together. If we didn't have our membrane like this, then we wouldn't have any cells, because it wouldn't hold together. Now remember, we talked about water and how we were going to talk about how if you dehydrate and you don't drink water, you're going to die. And the reason that happens is if you look at that cell membrane, it is filled with water on the outside and filled with water on the inside. Well, if you start to lose this water, the first thing that's going to happen is the water will probably go out here, because if this is drying up out here, the water will move toward there because there's less water, and eventually the inside will be dried out. Well, if the inside dries out, does the phospholipid care where it is anymore? No, the tails can hang out here now, and the head can turn. It breaks the membrane up. What keeps the membrane intact is water. That relationship of the phospholipid head and tail is what holds cell membranes together. As you dehydrate, as you start to lose water, the membrane falls apart. The tails no longer tuck in and hide from water, and the heads are no longer facing water. They just fall apart. As they fall apart, the contents can escape the cell and the cell dies. That's why if you wake up in the morning and you have a headache, it's because you're starting to dehydrate. That's one of the first signs of dehydration, is a headache. So a lot of times when you wake up with a headache, drink some water, you feel better. Okay. So here we have phospholipid bilayer that forms the cell membrane. And the relationship to water becomes crucial, because that's how the membrane holds its shape. If we start to lose water, we lose the membrane. If we lose the membrane, the contents of the cell escape, we lose the cell, and we start seeing tissue death. We can start losing all kinds of tissue when this starts to happen. This lipid bilayer is impermeable to most things except fat-soluble substances that enter the cell. So it's only things that are actually dissolved in fat that can enter the membrane. And those are things like nitrogen, oxygen, alcohol, fat-soluble drugs, and vitamins like A, D, E, and K. Let's take another look. So here's our cell membrane. Here's water surrounding the membrane. Here's water filling the membrane. And if we take a close-up look, you'll see here is a phospholipid, phospholipid, phospholipid all along, and they form a bilayer. Always the heads are facing the water on either side. The tails don't like water. Water doesn't like them, so they're going to tuck in the middle. and you have this beautiful, beautiful cell membrane. Here's another look. Here's our hydrophilic heads, hydrophobic tails. They're going to form this shape. Here it is. Do you see how it's just rows and rows of heads, and the tails are tucked in? On the inside, rows and rows of heads. There's something called a micelle that's kind of a whole cell, and there's not much on, think of like an M&M. This is just chocolate and chocolate coating. So there's no water on the inside, so it's okay that the tails just tuck in like this. These are micelles. But most of them have an interior and an exterior. Here's a nice look at a cell here. Do you see how the inside, again, all heads, and the tails are all tucked in here, safe, away from water. As long as this is full of water, as long as there's tons of water on the outside, it's going to keep its shape. We start to lose water, this thing's going to fall apart, and we get in trouble. That's why dehydration is so relevant. And here's what one looks like again. On top, we see all of these phospholipids. The heads all up in here, the tails here. Here's the next set of tails, and the next set of heads are inside here. But let's take another look at the membrane. It's not just the phospholipid bilayer. Do you see these other things on it? Well, when we talked about the phospholipid bilayer and we talked about things like permeability, if we look at the lipid bilayer, it's called the plasma membrane when we're talking about just the phospholipid bilayer. If we look at that, it's impermeable pretty much. It's not going to let much through. So this is pretty impermeable right here. In order to let things in and out of the cell, we have to have something else on this membrane. And what we have are proteins. There are various protein structures that you can see here. You see them? And some cholesterols that are on here. And these are going to play a really important part to the rest of the membrane. So the membrane is made up of a phospholipid bilayer that's pretty much impermeable. And then we have some cholesterols that are in there to help it maintain its shape. But then we have proteins, and those proteins are going to allow the permeability or the selective permeability. They're either going to act like channels, like little pores or holes through the membrane that things can just freely come in and out of, or they're going to physically take things that they selectively choose to enter the cell and move them through the cell. And we'll look more at that here now in just a second. The next thing is the membrane proteins. The phospholipid bilayer forms the membrane barrier of the cell. It's called the plasma membrane. Thousands of proteins are dispersed throughout the membrane, and they account for the selective and permeability of the membrane. If we look, do you see all these different things, just like we saw in this one right here? We see all these things on the membrane. Well, these are, for the most part, proteins. Like I said, there's also some cholesterols in here to hold the membrane together a little bit. But what we want to talk about now are these different proteins. There are three main types of proteins: integral, peripheral, and glycoproteins. You need to know this. The integral proteins go through the membrane from the outside to the inside. They transport substances into and out of the cell. And there are two types, channels and carriers. So when you talk about the integral protein, you need to know it goes from the inside to the outside of the cell, which means that it's able to move things all the way in and all the way out. And there are two types, channels and carriers. So let's take a look at that. Let's go back up here. If I have an integral protein, the first definition of that protein is that it goes from the inside to the outside. So here's the inside, here's the outside. You see how these proteins go all the way through the membrane and are able to transport things in and out of the cell? If you go here, these can't. And the ones that are just stuck up here like little gumdrops, they can't do it either. So if you look back at this one, do you see how this one goes all the way through the membrane from the outside to the inside. This one only goes through half the membrane. Do you see that? It only goes through half. And then there are some with these little antennas on them. The ones with the antennas are just floating on top of the membrane here. They're just floating on top. So here again, we have some that float on top. They're the ones with the cholesterol. This one isn't. This one's probably an integral. It's going all the way through. But again, you see some going all the way through from the outside to the inside, some that don't. They don't go all the way through. These are our three different types of proteins. The one going all the way through from the inside to the outside, we see it in all of these diagrams, are the integral proteins. And they are transporters, and there's two types, channels and carriers. All right. The next one is the peripheral protein, and it will be on either one side of the membrane or the other. And these have two functions. One, they maintain the shape of the membrane, and they work as enzymes. And enzymes are proteins that are going to cause reactions to happen. They're going to cause reactions to take place. So a lot of these chemical reactions that we look at are catalyzed by enzymes. Let's look at these diagrams. So right here, do you see how I have a protein that's only on one side of the membrane? Here's some more. This one's on this side, this one's on this side. See that? And if I come here, here's this one. You see? It's only on one side of the membrane, right here. And that is your peripheral protein. So this is your integral protein and your peripheral protein. Glycoproteins are involved in cell recognition, and they work as receptors. So if you're asked the question of what is the function of a glycoprotein, remember it has two responses. One, it's involved in cellular recognition. What does that mean? It means that it's out there, it's working as an antenna, and it's going to alert the cell to anything that's going on out there. Now, say for example, a bacteria is coming toward the cell. It is going to recognize that this is not a friendly cell, and it is going to call the immune system, like calling 911. We have an emergency, and the immune system then starts to send our immune cells, our fighter cells, out there to that bacteria to get rid of it before it can harm the cell. So these glycoproteins are monitors. They're sitting out there. They're like antennas, and they're sitting out there watching what's happening outside the cell. And if anything is going to jeopardize the cell or harm the cell, it is going to call the immune system. So they're very connected to the immune system, 911. That's what these cell recognitions do. You hear of transplants. We hear about it all the time, and we hear about how transplants either they're successful or they fail. And when you have a transplant that's successful, it is because these glycoproteins recognize those cells that are coming in as good cells, and they accept them. So you have a successful transplant. In the case that the transplant is rejected, these glycoproteins act to say, this isn't going to go. These are not friendly cells. They're going to call the immune system, and the immune system is going to fight them. So then you have a rejection, and you have to have that transplant removed. The other is receptor proteins. These receptor proteins whoops, get my drawing tool here. Here's the cell membrane, and they sit out here like little ready to receive something, and then a molecule will come and land in here, and then this receptor will take that inside the cell. So you have receptors, and these receptors are used mostly for hormones and drugs, so it relates very much to glandular systems of hormones that are going to make things take place in the cell. And drugs, different types of drugs. So if you're sick and your doctor gives you medication, they go to these receptor proteins and they're taken into the cell, and an activity is going to be stopped, or it's going to be enhanced, or something's going to, whatever it is that needs to be done is going to happen. All right, so we have three proteins, the integral proteins. They go all the way through the cell from the inside to the outside. They are the transporters. And there are two types, channels and carriers. The peripheral proteins, they stay on one side or the other. They have two functions, to maintain shape and work as enzymes. And then your glycoproteins, they're on the outside of the cell membrane. They're involved in cell recognition, and they're used as receptors. Cell recognition is monitoring what's going on outside the cell, and they work directly with the immune system. And receptors are the ones that receive these hormones and drugs and take them into the cell. Now, just one quick look back here at the integral proteins. We talked about them being channels and carriers. Channels are open. These are the ones we talked about that allow free flow of things into and out of the cell. And these would probably most likely be mainly for water. And then we have carriers. They are going to actually entrap whatever it is that we want to come in. These are the ones that are selectively permeable, and they will move them into the cell through the membrane and deliver them and drop them off inside the cell. So the channels allow a free flow of movement to specific molecules, mainly water, and the channel or the carriers are going to physically take things in or actually out of the cell. Okay, so they'll drop them off either in or outside of the cell. All right, so let's take a look at this one more time. Here is our integral protein. We know because it goes all the way through the cell, and we're going to have channels and carriers that are going to move things in or out of the cell. This is our peripheral protein. You know because it's only halfway through on either side. It could be here or here. And they are going to give the membrane stability, allow it to maintain its shape, and they're going to work as enzymes. And then on top with the antennas, we have all of these glycoproteins. And these glycoproteins are out here monitoring in cell recognition, working directly with the immune system, or they're working as receptors for hormones and drugs. Again, we see them here. This is your integral protein, your peripheral, glyco. Okay? Again, integral, peripherals, glycos. The ones with the antennas, glycos. Okay? All right. Here's another look at our various types of proteins and our phospholipid bilayer. These go into more detail about your different types of transporters and things that we already discussed. You can take another look at that. It explains channels and carriers a little bit, cell recognition, and enzymes. We've talked about all those things. All right, so now we're going to look at a cell. And remember, outside the cell is water, inside the cell is water. What you're looking at here, this yellow kind of glob, is the cell membrane. The blue on the outside, of course, is water, because that's where cells are. If we're saturated and hydrated well, we're going to be full of water.
The inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is,
now notice that the heads are all out here facing the water. This is what the outside of the membrane looks like.
It's surprisingly flexible. Push it and watch it move. Poke.
Now this part becomes really important to understand, because here's the heads, here's the tails, here's one phospholipid, here's the other. This makes up our bilayer, right? Notice that the outside of the cell is all water, the inside of the cell is all water. So we have our phospholipid heads facing water, and you see that these hydrophobic tails are tucked within the membrane. Now if something should happen or bump into the cell, what you're going to see is how it kind of breaks
Hard
up.
enough, and it might break and begin to regroup.
See how it broke up? But because of that relationship to water, it's going to go right back in place. These very hydrophobic regions of these tails are going to try to scurry away from the water as fast as it can, and these heads are going to try to face water as fast as they can. So that dynamic, that functionality is what causes the cell membrane to stay together. So watch what happens.
The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol.
So see how it just went right back in its shape. Water maintains the shape of the cell membrane. So if we're dehydrating, the cell membrane falls apart, we lose our cells. If we lose enough cells, we're going to lose tissue, whether it's lung, heart, brain, kidney, liver. It doesn't matter. We're going to lose those cells. Now we start to get sick because those organs can no longer function the way they need to. And if that happens, eventually, if you don't get some water, it's going to cause death.
And a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature comes in waste from the world out is for the cell. For instance.
Okay, now watch for a minute. You see this little molecule, whatever it is? It is going to go through an integral protein that is a channel, which means it's going to have free flow in and out of here. But if you look here and over in this one, you'll see that they can't go through. They have to be given permission to pass through. These are your selectively permeable transporters, the carriers. And you will see various other things happening too.
They receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick. Between the...
So now I'll show it to you one more time, straight through without me talking, and hopefully you'll get the visual.
The living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
When you're an entrepreneur, you have to risk everything. That 500 square.
All right, so now that we understand the cell membrane, let's take a look at what's inside. Well, when we look inside the membrane, here's our blue membrane. When we look inside the membrane, we have this whole area, and it's filled with something called cytoplasm, plasm being liquid or fluid, and cyte being cell, so it's cell fluid, cytoplasm, mostly water, but it has some other components to it, but for now, let's just think of it as water. It would be like a water balloon. The balloon would be the membrane, the water inside is the cytoplasm. Then the first thing we're going to look at that's within the cytoplasm suspending, it's called the nucleus. And we all know, I hope, that the nucleus is sort of the center, and the nucleus is very important because that's where the DNA and RNA are. DNA and RNA are found within the nucleus. Because the DNA is within the nucleus of a cell, it's referred to as the boss of the cell. The DNA is always the boss. The DNA is always going to win. Whatever is on that DNA for making proteins will determine the functionality of the cell. That's why they call it the boss. Here we have our membrane, the fluid inside the cell, the water in the water balloon is the cytoplasm, and the first organelle we're going to look at is the nucleus, the boss, contains the DNA and the RNA. So it's covered by a membrane. Now, once we're inside the cell, the membrane doesn't have to perform the same way, because the cell membrane itself has to determine what we're going to let from the outside into this cell. Once we've determined that everything that's inside the cell is pretty much what we want in there, we don't have to worry about that kind of stuff anymore. So the nuclear membrane, nuclear, will always refer to the nucleus. Anything that says nuclear, remember we looked at the nucleic acids, which are the DNA and RNA. Nuclear is always the nucleus. It's different from the cell membrane because it has a porous double membrane. It does not have the membrane proteins. And it's there to separate our nucleoplasm. Again, plasm is fluid, nuclei referring to the nucleus, fluid in the nucleus, nucleoplasm, from cytoplasm, again, cyto, cell, plasm, fluid, the fluid within the cell. So the nuclear envelope just separates the nucleoplasm, the liquid inside the nucleus, from the liquid inside the cell. Nucleoplasm contains chromatin. Now, when we refer to chromatin, we're talking about fragments of DNA. We don't really call our genetic material, DNA, until it comes together to form genes. So when it's in its tiny little pieces, fragments, it's called chromatin. And it has what's called a nucleolus. So the nucleus has another little organelle inside of it, and that little organelle is called the nucleolus. When we look at the nucleus, we have a membrane called the nuclear membrane. It differs in the cell membrane because it does not have proteins, and it's a porous plasma membrane, much like the lipid membrane or phospholipid membrane of the cell. So that's all it is just the phospholipids, but it's porous. It has holes in it. If you think of like a wiffle ball, those little plastic balls that your kids play with, only the holes are smaller, that would kind of be what the nuclear envelope looks like. And it's there to separate the cell fluid from the nuclear fluid. Within the nucleus, we have a lot of chromatin, which are fragments of DNA, and what's called the nucleolus. The nucleolus is a small organelle, and it's in the nucleolus that we have the RNA. So in the nucleoplasm, we have our DNA, that's in the nucleus, also a little nucleolus, and the nucleolus is where our RNA is. If we look here, we can take a closer look at that. Now notice that your nuclear envelope or membrane, you call it either one, I don't care which one you call it, whether you call it nuclear envelope, nuclear membrane, makes no difference to me. But if you look carefully, do you see the holes? These are the pores that I was talking about that allow things in and out of the nucleus. And we don't need those membranes or the proteins anymore because we already know that what's in the cell is what we need in the cell. So now we just have pores that allow movement of things that we need inside the nucleus or we want to remove from the nucleus. And we'll get into that later on when we start talking about protein synthesis, how we make proteins. If you look here, the light lavender color, that's our cytoplasm, or nucleoplasm. The lavender is the nucleoplasm, so that's our beautiful liquid within the nucleus. And you'll see all these purple strands. All these purple strands that you see in here, that's the DNA. Now, if you look at this ball that's inside the nucleus, that is the nucleolus. And the nucleolus is filled with RNA. So these fragments of DNA in the nucleus are called chromatin, and inside the nucleolus is where we find RNA. All right, nuclear envelope with pores, the light lavender, nucleoplasm. In the nucleoplasm, we have all these stringy fragments of DNA called chromatin. Here's our nucleolus, and within the nucleolus, we have RNA. Next is the cytoplasm, which we've already kind of talked about. It's the fluid that fills the cell. You'll also hear it called cytosol. Cytosol, cytoplasm makes no difference. They're the same thing. They suspend organelles in the cell. They're made up of water, salts, enzymes, fatty acids, sugars, amino acids, and waste products. These things that keep the cell working. So at various times, all these things are going to be moving around the cell, doing their work, doing what they have to do. So. All right, now we're talking about movement. When within the cell, you'll see in your textbook there's two types. There's passive transport and active transport. And passive transport is just what you think. It's very easy, it's graceful and gradual, and it does not require energy. It doesn't require energy. We don't use ATP for it. Movement of substances from regions of higher concentration to lower concentration. Now, when we talk about any type of diffusion, this is the definition. We'll get back to that in a minute. It doesn't involve energy. It's free flowing. So think about walking. Maybe you're walking with a big crowd, and you're all kind of moving in the same way, and they just kind of help you move along. Actually, it's just kind of a free flowing. Everybody's headed in the same direction. It doesn't require a lot of energy. There are 4 types of passive transport: diffusion, facilitated diffusion, filtration, and osmosis. Now, you're going to have to know the definitions of these, so you're going to have to memorize this. movement of substances from regions of higher concentration to regions of lower concentration. And this will change a little bit for each one. Now, in the case of diffusion, it's movement of substances from a region of high concentration to a region of low concentration through space until it reaches equilibrium. What I mean by that is it's going to move through a space until that entire space is evenly filled with it. So, for example, take like an air freshener. If you have a can of air freshener and you stand on one side of the room and you spray it, it won't take long before the whole room smells like that air freshener. And you can actually, it's so concentrated when it comes out of the can that you can see it. You can see these little droplets of liquid, whatever it is, that's coming out of the can. Now, once that happens, that concentration, those little particles that you saw start moving through the air in that room. And before long, it reaches equilibrium, which means it doesn't matter if you're on the roof or the ceiling or the floor or one side or the other or in a corner or in the middle of the room, the whole thing is going to smell the same. And it's because those little particles that you saw come out of that can have evenly distributed throughout that entire space. It works a lot the same way like with Kool-Aid. If you put a package of Kool-Aid in water and stir it, pretty soon that whole thing is equally distributed, and every single person who gets a glass of Kool-Aid, it's going to taste the same. One's not going to be more watered down than the other. They're all going to taste the same. It's because of diffusion that the Kool-Aid moved through until it reached equilibrium. Same thing happens with air fresheners. It's all going to smell the same. Pretty soon the whole room is going to smell the same because it reached equilibrium. Definition for diffusion, write it down. Movement of substances from regions of high concentration to low concentration through space until it reaches equilibrium. That last part is the only thing that's going to change for all four of these definitions. Facilitated diffusion, movement of substances from regions of high concentration to low concentration with help until it reaches equilibrium. And what I mean by with help, now if you think of putting water in a coffee pot and it hits the filter with the coffee granules in it, that filter is going to allow things to pass through. But when you see the coffee passing through, now the water's mixed with the coffee granules and you can see it all come out and it's going to taste the same, right? The whole pot of coffee is going to taste the same. So the filter kind of helps diffusion. It helps diffusion. That's why we say with help. Facilitated diffusion is the movement of substances from regions of high concentration to low concentration with help until it reaches equilibrium. And coffee is a perfect example of that. Oh. No, it isn't. I messed up. That's filtration. Sorry. The one I gave you is filtration. I wasn't thinking about facilitated diffusion. I'm sorry about that. Go back and delete that. It is not facilitated diffusion. That is filtration. Now I will tell you about facilitated diffusion. Facilitated diffusion is a lot like filtration in a way, except it refers more to those selectively permeable things, those proteins that will select what's going to go in and what's not, and those channels. If I have water diffusing in and out of a cell and I have an open channel, do you see how the channel is helping the water get from one place to the other? That would be facilitated diffusion. Movement of substances from regions of high concentration to regions of low concentration with help until it reaches equilibrium. Filtration, movement of substances from regions of high concentration to low concentration through a filter until it reaches equilibrium. So you might have to go back and re-listen to that because I got things mixed up. Osmosis is movement of substances from regions of high concentration to lower concentration through a semi-permeable membrane until it reaches equilibrium. Now we'll take a look at what I mean by that. Make sure you know those 4 definitions. All right, when we look at passive transport, we're looking at diffusion, simple diffusion. We're going to move from a region of high concentration to a region of low concentration until we reached equilibrium. Facilitated, we have HELP, right? So for facilitated diffusion, we have HELP. Diffusion is simple. Molecules are spreading from high to low concentration until they reach through space until they reach equilibrium. Kool-Aid, perfume, air spray. Facilitated diffusion from a region of high concentration through low concentration using HELP of some sort, but we're not going to get energy from it, but it's just using HELP until it reaches equilibrium. Filtration, movement from a region of high concentration to low concentration through a filter until it reaches equilibrium. Osmosis is diffusion through a semi-permeable membrane until it reaches equilibrium. So what happens in this case right here that we're looking at this, if we have a tank of water, okay, and we put a membrane, a semi-permeable membrane, so it would look like this, it would have little holes in it because it's semi-permeable. So we're going to allow things to pass through it. And over here we have water, and I fill this tank over here with water. If I leave that, what's eventually going to happen? Well, this water is going to come down and it's going to reach equilibrium, right? These both sides are going to be the same. Now, if I dump a bunch of salt or some sugar or something in here, what is going to happen to that? Well, eventually it's going to move this way until both sides have the same amount, right? So the end result would look something like this. It looks something like that. It's going to be even. So it's movement from a region of high concentration to a region of low. If you look here, is there any salt over here? There's no salt. That's low concentration. Here's high concentration. Here's the high concentration of salt. It is going to move to the area of low concentration, and this salt is just going to keep moving over here until it reaches equilibrium, until they're the same on both sides. Okay? Region of high concentration to a region of low concentration through a semi-permeable membrane until it reaches equilibrium. If you have any problems understanding what I mean by that, make sure you let me know. Now, active transport is a little bit different. Unlike passive transport, active transport works against diffusion. It moves molecules uphill against a gradient, and it requires energy. Now, I use the analogy of going with a crowd. Let's say that you're at a concert and it's a hot ticket and there are 8,000 people at this concert. When the concert is over, everybody gets up and tries to flow out of this venue. If you're going with the crowd, they're pushing you along, they're helping you. It's a passive transport. But what happens if you're reaching the door and realize that you left your wallet or your purse where you were sitting? You're going to try to turn around and work your way back to your chair to see if you can find your money, right? So you turn around and all of a sudden you have 8,000 people coming in the opposite direction of you. What happens when you're fighting your way through that crowd? You're using a lot of energy to push your way back. A lot of energy. Think of that as active transport. Passive transport is when you're all going in the same direction. You're practically floating because the energy of everybody going in the same direction, it doesn't require much energy on your part at all. But if you turn around to try to get back up to your seat and you're trying to work your way through all of those people, you're using a tremendous amount of energy. That would be active transport. You're moving against an uphill gradient. You're fighting against the flow. There are two types, endocytosis and exocytosis. When we talk about passive transport, we're talking about the movement from a region of high concentration to a region of low concentration through something until we reach equilibrium. And we have diffusion, facilitated diffusion, filtration, and osmosis. Active transport is the other way around. We're fighting. We are using energy. We're fighting that crowd. And there are two types, endocytosis and exocytosis. Endo means that we're bringing something in to the cell. So if I have my cell, endocytosis, I'm bringing something in to the cell. Endo, in, within, and cell is site, so I'm bringing something within the cell. Exocytosis, I'm taking something out of the cell, okay? So I'm using energy to move something into the cell, or I'm using energy to move something out of the cell. Active transport, I'm using energy. Endocytosis, exocytosis. Endocytosis, I'm using energy to bring into the cell. Exocytosis, I'm using energy to take out of the cell. Here's a look at exocytosis. When we look at exocytosis, there are three types of exocytosis: pinocytosis, phagocytosis, receptor-mediated. All terms you need to know. Pinocytosis means that we're taking in water. Phagocytosis means we're taking in solids. And receptor-mediated is those, remember we talked about the receptor proteins that are hooked onto hormones and drugs? That is how we bring hormones and drugs into the cell. It's called receptor-mediated cytosis. Receptor-mediated cytosis. So when we talk about active transport, we're using energy, two types. Endocytosis means we're bringing things into the cell. Exocytosis, we're taking things out. When we talk about endocytosis, we have three types: pinocytosis, phagocytosis, and receptor-mediated cytosis. Pinocytosis, we're bringing water in. Phagocytosis, we're bringing in solids. And receptor-mediated cytosis, we're bringing in those hormones and drugs that are attached to the receptor proteins. So if we look down here at what a picture of that looks like, do you see how it comes onto the membrane and the membrane engulfs it? Do you see that? The membrane engulfs it. And it forms this round bubble of membrane. And these round bubbles of membrane are called vesicles. They're called vesicles. They're these pieces of membrane that pinch off because they're carrying things throughout the cell. A vesicle is a transporter in the cell. Vesicles are what take things to their destination. And when we're talking phagocytosis, we're taking in solid particles. We're forming a vesicle from the membrane around this particle. It's going to pinch off, and the vesicle is going to carry whatever this is to its destination within the cell. Pinocytosis, it's how we bring water in. Water, the membrane is going to pinch or fold around it and pinch off to form a vesicle that is now going to deliver this water wherever it needs to go. And the same with, here's your receptor proteins. You see them up on the membrane. They're hooking up with various hormones or drugs. And now you can see that this membrane is going to form a vesicle. And you see how now it's engulfed the receptor proteins with whatever it is they need to bring into the cell. Now they will deliver them, and this will go back up to the membrane, and those receptor proteins will be put back up on the membrane. Exocytosis, there's only one thing to know, and that's exocytosis. It's how we get things out of the cell. Now, as we learn more about the organelles of the cell, you're going to find out that there's Golgi apparatus, there's endoplasmic reticulum, a lot of things here that also have membranes that produce things. Now, when we eat those proteins, lipids, carbs, they get broke down in the digestive system. They're carried through the blood and delivered to cells. Cells use them to make various things that the body needs. So once they get inside the cell, the vesicle is going to transport them to different places where we're going to actually use them. Once we have made the products that we need from them, or if we have waste products, these different organelles will export them by forming vesicles again from the membrane. They'll pinch off, and these vesicles now will go up and fuse with the membrane. See how this is the vesicle here, and now it's opened up, and what's inside of it can now be delivered outside the membrane. So that's exocytosis.
Cells also use cells.
Okay, so now we're going to look at this membrane again that we were looking at. We're going to look at how this active transport takes place, endo and exocytosis.
Cells also use energy to transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior. In the opposite process, called exocytosis, the sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents. Cells
Okay, so that's how we transport things in and out of the cell, through passive and active transport. Let's take a look at the rest of what's in the cell. Now, you have some interesting labs this week. One is you're going to turn your phone into a little microscope. And once you do that, you're going to start looking at cells. And the first cell we're going to look at is the animal cell. And it's going to be human. It's going to be your cells. You're going to take them from your cheeks and stain them with some iodine. And when you see them, this is kind of what they're going to look like. Not as close up as this, but you will have something that looks like this. You'll see your cell membrane, a bunch of cytoplasm, and this big egg-shaped thing is going to be your nucleus. And when you look away from this, you see a whole bunch of stuff in here. You don't know what it is, but there's a bunch of stuff in here. And there's this, and there's these things. And you see this dark stuff that's surrounding the nucleus. These are all organelles. These are all things in the cell that cause it to have function, that are going to take those proteins, lipids, and carbs, and they're going to do things with them. They're going to make things out of them for the body to use. Structural units inside the cell that have a specific function are the organelles. They allow the cell to function properly. When you're viewing a cell under a microscope, the cytoplasm is clear with scattered specks, and those specks are the organelles. So all these little things that you're looking at are actually organelles in your cell. Activities of a cell occur largely in the cytoplasm, where nutrients are received, processed, and used in metabolic reactions. What do we mean by metabolic? It's a series of processes by which food is converted into the energy and products needed to sustain life. So when we're having metabolic activity, we are taking these things apart and then we're putting them back together for things that we can use. For example, when you eat a steak, we're going to take it apart into amino acids, and then we're going to get those amino acids down to the cell, where the cell is going to start producing proteins that we need from it. So that's metabolism. Organelles of the cytoplasm have a specific function for these life-sustaining metabolic activities. The first one is called the endoplasmic reticulum, the location for the synthesis of protein and lipid molecules. The endoplasmic reticulum is always right around the nucleus. So do you see this dark region right through here and right through here? That is going to be the endoplasmic reticulum. Of course, all you can see is maybe a shadow of it. In these, you'd need an electron microscope to actually see it. But even when we looked up here and we were talking about our nucleus, did you see this structure in front of it? This structure in front of it is the endoplasmic reticulum. You're always going to find endoplasmic reticulum around the nucleus. And there's a real important reason for that, and we'll find that out when we go to make proteins. The endoplasmic reticulum is this very layered, structured thing, and it's divided. There's two different types of endoplasmic reticulum. We have a rough endoplasmic reticulum, and we have what is called a smooth endoplasmic reticulum. When we go back up here again at this picture that we were looking at the nucleus, do you see all these little dots on the endoplasmic reticulum? See, the endoplasmic reticulum is this kind of foldy shaped thing. We'll look at more pictures of it and you can see it more clearly. But do you see all these little dots on it? These little dots on the endoplasmic reticulum indicate that it is rough endoplasmic reticulum. Areas of the endoplasmic reticulum that are covered with ribosomes are rough endoplasmic reticulum, the site of protein synthesis. Areas of endoplasmic reticulum that don't have the ribosomes, those little BB dots, have a smooth appearance, and they're smooth endoplasmic reticulum, the site of lipid synthesis, absorbing fats and breaking down drugs. So let's take a look first at the rough endoplasmic reticulum. This is what it looks like. You can see that it's wrapped around the nucleus. It has all these little dots on it. These little dots are called ribosomes. Now, this organelle has a very specific function. When we look at the rough endoplasmic reticulum, it is the site of protein synthesis, which means this is the building inside the cell that we're going to build proteins in. It doesn't build proteins, it is the place that we build proteins. So the first thing you have to know, rough endoplasmic reticulum, it is dotted with ribosomes. It is the site of protein synthesis. Now it's the ribosomes that actually assemble the proteins. So these ribosomes are these little things all over the rough endoplasmic reticulum that will actually take amino acids and create proteins out of them. So the rough endoplasmic reticulum, site of protein synthesis, these little dots are the ribosomes. They are the ones who actually assemble proteins. When we look, here's rough endoplasmic reticulum. Almost always, you can find chunks of it in the cell and other locations, but most of it is surrounding the nucleus. And look closely. Do you see we have some that does not have ribosomes on it? That is smooth endoplasmic reticulum. This one here, you see it's just coated in ribosomes. This is our rough endoplasmic reticulum, always found with right around this nucleus here. Now, the smooth endoplasmic reticulum is also a factory, and this is where we take care of drugs and our fats and things. So drugs and fats and things are taken care of in the rough or smooth endoplasmic reticulum. Rough endoplasmic reticulum is the site of protein synthesis. The ribosomes are the assembly workers that go in there and actually create protein. Here's another picture of that. And again, here's our nucleus, our nuclear envelope. You can see that it's connected, very connected to this rough and smooth endoplasmic reticulum. The rough endoplasmic reticulum, you see the ribosomes. Smooth, you do not. Smooth is the site of lipid synthesis. rough is the site of protein synthesis, and the ribosomes are the ones who actually do the assembly work. They assemble the proteins. The next one is called the Golgi apparatus. They're stacks of flat membrane sacs called cisternae. They are responsible for packing and shipping. They also do quality control. So there's 3 jobs of the Golgi apparatus. It packs, ships, and does the quality control of the products, which are proteins that are made in the cell before we ship them out to be used. It refines, packages, delivers proteins synthesized on the rough endoplasmic reticulum. The proteins enter the Golgi apparatus. They pass from layer to layer where they're modified. And when proteins reach the outermost layer, they're packaged in bits of Golgi membrane that bud off and form transport vesicles. When we looked at the vesicles, when we were looking at exocytosis and endocytosis, we saw those vesicles that are being made. In exocytosis, remember I told you that there are organelles that produce things and they're going to ship things off to wherever we need them, and the Golgi apparatus is one of those. So it is one of those that actually packages these proteins and it forms vesicles around them, and now those proteins can be delivered anywhere. This vesicle travels to the cell membrane where it dumps its contents, or some of these proteins can be delivered within the cell. Here's your Golgi apparatus. Here's what it looks like. You see that it looks like a bunch of folded pancakes. But the real telling thing is that you see all the incoming traffic and all the outgoing traffic. These incoming vesicles are bringing proteins into the Golgi apparatus. The Golgi apparatus is going to make sure that these proteins are absolutely perfect. They're going to refine them. They're going to QC them. They're going to make sure they're perfect. If the proteins aren't perfect, they're going to reject them. They're not going to go to the body. So now, once these proteins have passed inspection, they're going to be packaged in vesicles, and they're going to ship to the membrane or throughout the cell, wherever these proteins need to be delivered. Golgi apparatus, it refines, packs, and ships proteins. And here's another picture. Here's a picture. I like this because you can see it all put together. Here's the nucleus. Here's the rough and smooth endoplasmic reticulum. From the rough endoplasmic reticulum, do you see these vesicles that are taking the proteins that were produced here and dropping them off to the Golgi apparatus where they're going to refine them. And once they're good to go, they're going to pack them up in vesicles and ship them off wherever they need to be. Next are the vesicles, and we already talked about that. The membrane sacs that form when a portion of the membrane breaks off transport vesicles for various materials. It takes substances into and out of the cell, and it includes vesicles formed by the Golgi apparatus, which we just saw, the cell membrane, and we also saw that we get these vesicles pinched off from our rough endoplasmic reticulum. Also, smooth endoplasmic reticulum sends out our lipids and things, and so you can see that all of these membranes can make vesicles to transport their products throughout the cell and to the membrane to go outside the cell. And here's a nice depiction of just what a vesicle looks like.
This is a cell that secretes them. This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane. They leave the cell instead by a process known as exocytosis. After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus. Their membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As membranes merge again, the contents leave the cell without actually crossing the plasma membrane. This process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis.
Our next organelle is the mitochondria. It's a kidney bean-shaped organelle. It has 2 layers, and it's kind of important to remember that it has 2 layers because once we get into cellular respiration, you're really going to have to understand the structure of this organelle. It has an outer and inner membrane. You see the regular outer membrane. Now, this usually is just covered in a membrane, of course, but it's cut away in this diagram just so we can see the inner membrane inside. Do you see this cristae formation of membrane on the inside? That's the inner membrane. So we have an outer membrane and an inner membrane, and we will get to know this organelle really, really super well. This is where we make energy. This is where we make that molecule of ATP, adenosine triphosphate, that I've told you about. So when we eat those carbohydrates, this is where we want them to go. And again, we can also use fats and proteins for energy, but the body will prefer to use the sugars first. Here's another view of the mitochondria. We see the outer membrane, and you see this inner membrane? And look between the inner membrane and outer membrane. Do you see that there's no water there? It's a space. The water this is like an inner tube, and it's holding water on the inside. You see that? This becomes really important when we start talking about cell respiration. But this inside where the water is, the pool here, is called the matrix. That's called the matrix. And here's our inner membrane and our outer, and the space in between is called the intermembranous space. We'll talk more about that later, but this gives you a nice introduction to the mitochondria. Here is the mitochondria as you would see it under a nice electron microscope. Then we have our lysosomes. When you think of a factory, we have all these areas where we build and create things, and that's like the endoplasmic reticulum and the Golgi apparatus, very busy parts of the cell. And we also need to have a cleanup crew. We have to have a maintenance crew, and that's where these lysosomes come in. They're tiny membranous sacs that contain enzymes that can degrade and recycle various molecules. So there's going to be waste products. There's going to be things that we don't need in that cell. And these are the scavengers. They're the ones that are going to come clean everything up. So the lysosomes are the cleanup crew. We also have peroxisomes. Now, they're like a lysosome. They also have enzymes, but they are very specific to toxic substances like alcohol, and they're going to prevent it from doing any harm. Now, the place that we find peroxisomes are the liver and kidney, because the liver and kidney are the ones that process these toxins. There's a lot of things that are toxins. It's not just like alcohol and drugs and stuff like that. I mean, there's parts of coffee that are very toxic. Even if you take an aspirin, it's very toxic, even though it's over the counter. If you eat enough of these, you can overwhelm your liver. But the peroxisomes, they're the biohazard crew. They're the ones who are going to come in and take care of toxins. Once we lose peroxisomes in our liver, people who may drink too much alcohol, they start to have cirrhosis of the liver, and eventually they can die from it. And that's a conversation for anatomy, but that's where this peroxisome comes in. Now we have things that aren't necessarily organelles, but they are called cytoplasmic structures. These are structures that have important functionality, but they're not really considered organelles. The first one is what we call a centriole. And you can always tell you're looking at a centriole because it either looks like this, a kind of bunch of rods put together, or it might have this odd star shape to it. When you see anything like that, that is a centriole. And the only thing you have to know right now about the centriole is it's important in cell division. That's all you need to know about it. Then we have things like cilia and flagella, and these are just little things on like some bacteria cells and even some little plant cells and protozoans that help with movement. And even in the human, we have cilia that lines like our intestinal tract that helps move digested food particles through the intestine. But they're these little hairs that move things along. And then we have flagella, and these are little whip-like structures, and a lot of protozoans and bacteria have whips to help them move. In the human, we can relate it to sperm. Sperm have these flagella. The other thing are called these microfilaments and microtubules. They're rods and tubules that support the cytoplasm, give structure to the cell, and aid in mitosis. So again, what you have to know about this is pretty much what you have to know about the centriole. It is going to, well, aid in cell division, but it also gives structure to the cell. Let's take a look now. If we look again, here are the organelles, here are the structures, everything that we just talked about. Here's our cell membrane. Here's the Golgi apparatus, and you can always tell because it's got incoming and outgoing traffic. See that? The smooth endoplasmic reticulum because it doesn't have ribosomes on it. The rough endoplasmic reticulum, you can clearly see the ribosomes. Don't pay attention to this because we don't have it in our cell, but in the animals, the ones we're looking at. Anyway, these are the mitochondria, and you can tell because they always have them sliced open where you can see the inner and outer membranes of the mitochondria. Now here are the centrioles. Remember I told you those rods or the star-like structure are always going to be centrioles, and that's what we're seeing here. Here, if we look real close, we're also seeing another chunk of rough endoplasmic reticulum, we know because it has ribosomes on it. Now when it comes to these dots, it's kind of hard to tell lysosome from a peroxisome. So if you're asked to identify these, just identify one as the lysosome, one as the peroxisome, and you will know what it is that you need to know. All right, summing up the cell, think of it as a factory. You walk up to the building, that's the cell membrane. The receptionists are the proteins. They're going to decide who comes in and who goes out. And the factory floor is the cytoplasm. The boss is the nucleus. He's going to control and regulate the activity. Our power plant, the mitochondria, that's who produces our energy. The assembly line are the rough and smooth endoplasmic reticulum, and they are going to be the site of the assembly of proteins and fats. The workers are the ribosomes, the packing, shipping, and let's not forget the QCing apparatus is the Golgi apparatus, and it's going to inspect the product for flaws, clean them, wrap them, and ship them. Who cleans up the mess? Our maintenance crews, the lysosomes and the peroxisomes. The peroxisomes are going to dispose of harmful products. The lysosomes clean up the trash and force things out of the cell. And there is the last look at that. So make sure that you understand the animal cell, you know the physiology of that cell membrane and its relevance to water, and that you know all of these organelles and structures, you know what they are, you can identify them, and you know what their functions are.